Thanks for having me and also making sure that I'm audible. I'm Sebastian Fürthauer. I'm actually a local from the other big one of the other big universities in town, from TU Wien. And me and my group are theoreticians that worry about things like you see here, materials that come from cells, from cell biology, that are made out of polymer filaments and actuated from within by specialized molecules, so-called molecular motors, that can consume the chemical energy from within cells and do mechanical work. And we are trying to sort of understand what this means for the large-scale physical properties of these materials. And there's like two classes of systems that we are interested in. One that I will not be talking about today, which are collectively beating cilia, which are whip-like appendages of cells that generate fluid flow within your lungs, within your kidneys, within your brains, but also help like little bugs like paramecium or something to propel themselves through their environment. And the second one that I will talk about today are these kinds of systems which come from the cell cytoskeleton. Since I'm the first speaker here talking about systems from biology, I will start with the assumption that you know nothing, but are infinitely smart, so will understand everything that I say. To make sure that this is a good approximation, please interrupt me with questions whenever we are sort of reaching the limits. I'd rather not finish the talk than sort of lose you halfway. Okay, so the plan is we'll first give you sort of a brief, very opinionated introduction about the cytoskeleton. Then I will introduce you to key concepts of how we think we can derive continuum descriptions of such a complex active material. I will do that mainly at the blackboard. Then I will return to my slides and show you a few examples that make us hopeful that sort of the techniques we developed are actually useful in comparison to real experimental systems, both in vitro, in the lab, and in vivo. So what is the cytoskeleton? The cytoskeleton is a network of polymer filaments that spans our cells. And these polymer filaments are connected by cross-linking molecules. And together, this type of material sort of acts as the bones and muscles of our cells. They sort of set cell shape and endow the cell with the property to deform itself actively and to work to its organelles, so to its inside and to the outside world. Instead of giving you the classic textbook type of view of that, I thought I would just show you sort of the first 30 minutes of the life of an organism, of the life of a cell, and sort of highlight some of the key features of the cytoskeleton. So what you see here is a worm. That is C. elegans. If you ever reach to the fruit bowl, you eat these guys. They live in soil and on vegetables. They're like a millimeter long and like 100 micron thick, transparent, hermaphrodite worms. And the structure that do we have a stick or something like that? On the other side. This is kind of brutal, but it will work. The structure that you see here is actually the gonad of the worm. There's two of them, one on this side, one on that side. And that is what, oh yeah, that is much more elegant. Yeah, ask questions. And this guy or girl is a hermaphrodite, so it sort of produces its own eggs. They sort of are produced here over the course of roughly a day. The new eggs grow. Here is the spermatheca, where it produces its own sperms. The eggs are fertilized, and this here is really the one-cell stage embryo that you will be seeing. In the next few slides. So, and that's basically what happens the first 30 minutes after it's fertilized. In green here is labeled histones, so that is basically a marker for DNA. And what you have seen in the beginning of the movie, and we'll see again as it loops, is sort of the female pronucleus, so the female genetic material starting out in the middle of the cell, meeting the male pronucleus that just came in with the sperm, going to the middle of cell, dividing, going to the two daughter cells, and the cell itself sort of deforming. And just because it's easier to follow, I put you sort of a plot of time scale, like a time plot here of the key events that happen. But basically, if you want to understand that, what you want to understand is a series of mechanics problems, of self-organized mechanics problems, where networks of filaments self-organize to, in a very stereotyped way, move around the internal organelles of cells and give rise from one cell to two cells and more. And it's hard to understate the importance of this process. To go from one egg cell to any of you have to manage to do this without too many mistakes, 10 to the 13 times. There's about 100 times more than there are stars in the Milky Way. The number of cells in each of your bodies that divide right now that do this is about the same as the number of stars in the Milky Way. And if this goes wrong too often, terrible things happen. So there is, of course, reasons why we want this. So there's obvious reasons why we would want to understand this. And so the two systems that sort of stick out in this movie to me is the one that forms here, which is called the mitotic spindle, which is responsible for the division of chromosomes. And then, which I will discuss a little bit later, the cell cortex, which sits at the surface of the cell and sort of is responsible for its large-scale deformations. So let's start to focus in on the mitotic spindle. That's sort of the textbook view of this. There's two specialized structures here called the centrosomes, which come with the male pronucleus. There's some polymer filaments that sort of manage to collect chromosomes in the middle of the cell, then pull them apart and divide them. So that's the textbook view, but the filaments that you see in there are so-called microtubules. So these are polymer filaments, so something that you're familiar with, but these are quite special polymer filaments. So unlike many materials that we would have in the lab, which sort of you get to polymerize by basically having an excess of monomer in solution, thus just using the chemical energy that comes with that to grow and get an exponential length distribution out of that. These filaments are, first of all, structurally polar, so their plus and their minus ends are different, and they tend to polymerize more from their plus end and less from their minus end. This polymerization is not an equilibrium process. It is also coupled to the hydrolysis of a chemical fuel, GTP. So this is the first thing that makes this material very different from equilibrium material that we would have in the lab. Each of these filaments consumes energy in their growth and shrinkage process. It is structurally polar. That means there's a plus and a minus end that I can then define. And such a microtubule has a persistence length, so that is a measure for the stiffness. It basically measures the distance over which thermal fluctuations would be sufficient to make one end forget the orientation of the other of several millimeters. So that is huge on the scale of a cell. They're basically rigid rods. However, in spindles, they're very transient structures. The typical lifetime of these things is like 20, 30 seconds. The exact number is then cell type and context specific, but they're not very long-lived things. Okay, just to give you a sense how these C. elegans spindles look in a real cell, what you can do, and what our colleagues in the Rademann lab and in the Müller-Reichert lab have done, you can take one of these C. elegans embryos that you saw before, freeze them, embed them in plastic, cut this thing into little slices that are about 75 nanometer thick, and then do electron tomography on it. reconstitute the whole thing. That's sort of the type of data set that you get out of this. What you see here is the mitotic spindle that you saw 2 slides ago under light microscopy moving here in a very dead state. But you can see each one of these little polyfilaments. Each of them is a little microtubule. You can see the chromosomes in the middle. And you can see that really the structure that we're thinking about is indeed, as I claimed, a cross-linked polymer network. Just because it's so beautiful, I'll also show you the zoom in. You can even figure out which of the microtubules is connected to which of the chromosomes and ask questions about chromosome segregation and centering data sets like this. The second thing, yes?
Can you go back? Yeah, just before you say that these the persistent length was in the literature, yes. You see that there are some kind of twists. I mean,
so the persistence length that I quoted is thermal fluctuations with room temperature. Here you have tons of non-equilibrium processes. The first of one, already mentioned, is that the filaments themselves consume energy while growing and shrinking. So they exert forces which are many kTs, like 1 GTP is 40 kT, is that correct? So there's forces that are much stronger than thermal fluctuations from the growth process. And then there's molecular scale motors, which again at each step consume 1 ATP, which again gives you a manifold of thermal fluctuations. So while it is true that thermal fluctuations can't bend these things, a lot of intracellular force generators can, and can very easily. Okay, the second thing that I want you to know about these networks, this is now another spindle. This comes from a frog, and it's much bigger than the C. elegans one. So it's like this 50 micron shaped football. And there's 2 experiments on the same structure at roughly the same time. The difference is, to see these things under the microscope, what experimentalists do is that they spike in fluorescently labeled tubulin, which is the monomer of microtubules. And here they spiked in a lot, so the whole structure lights up. And this is a photo, but if I showed you a movie, you would just see this in steady state just flickering about. What you see on the right-hand side is an experiment where the experimentalists just gave a very little amount of fluorescent protein. And so what you now see is that every once in a while, a microtubule in its growth shrinkage process picks up 1 little fluorescent monomer. That's the dots that you see here. What I want you to appreciate, so the structure is rotated by 90 degrees between these 2 movies, but otherwise it's the same structure. But what I want you to take from this movie is that each of these dots moves. So each of the microtubules in the structure is in permanent motion. So not only is this network of filaments, which on the EM reconstruction looks basically like something that we could build, like something like the Eiffel Tower, but not only do the individual filaments turn over very rapidly on the time scale of the existence of these things, but also each of the filaments is in permanent motion. So really what we are seeing here is maybe more akin to an ant trail than to a structure that we would build out of metal trestles.
Yes. Is this like an active motion made from the cell, or is it just a passive motion? I have my microtubular structure in the cell. For some reason, the plasma.
So I will come to this, but this is an active motion. We can identify the motor that is responsible for it, and our experimentalist colleagues can kill it off, and these dots will stop. So basically everything here is active. So so one of the key points that I tried to get across now is that in all aspects of the problem, you're very far away from thermal equilibrium all the time. Okay, so if you see something move at low Reynolds number, I guess 0 Reynolds number is that a word that confuses people? Okay, at 0 Reynolds number, that means that the total force on this filament, F equals ma, is always 0. But that means that the drag of each of these filaments with respect to its environment always has to be balanced instantaneously by an active force. And so that will sort of be one of the key insights that we will use later on, is that whatever we do in these systems, the total force on each filament is always zero. If I see it move, there's an instantaneous force that balances drag with the network and the outside world against active forces. I sort of already gave away where these active forces come from. On one hand, they can come from the fact that these filaments grow under consumption of energy, but more importantly, they come from guys like this. I stole this from a movie that you can find on the internet that's called The Inner Life of the Cell. People have strong opinions about this. I encourage you to look at it, but also read all the comments, because that really points you towards like what like people in our field are worrying about right now. But what I want you to see for now is like this example of, in this case, what's supposed to be a kinesin molecular motor that carries a vesicle. The way you should think about these molecular scale motors, it's really like this, like little guys stepping along the filaments. One thing that this movie grossly misrepresents is how noisy the environment of the cell is. So most likely these guys will not go as nicely and coordinate like this, but more like their little dance, fluctuate up and about with a bias towards one direction. So these molecular motors consume ATP and convert this into mechanical work. And importantly, in our spindle context and in many contexts that we will discuss today, these molecular motors can couple several filaments. So unlike the video that I showed you before, where this transports a cargo, the cargo can very well be another filament. And so in the spindles, since I will use these words later on, there's like several flavors of molecular motors that are important. One being kinesin-like molecules that are plus-end directed motors. The other one being dyneins that are minus-end directed motors. And the kinesins that I will talk about connect two microtubules and basically slide them past each other. And they are actually responsible for this flux. The dynein-like motors have one end that's just statically attached to a microtubule minus end, and they reach into the network and pull towards the minus end of whatever microtubule they find. So the fact that you have several flavors of microtubules in there is actually important for the morphology and the function of the network. So that's the healthy frog spindle you saw before. What happens here is if I kill off the dyneins in the system, you get to this much more flared out structure that people call barrel-shaped. That is what happens if I kill off the kinesins. You get to something that basically looks like a hedgehog. The point of this is that what flavor of motors are in the network really shapes the emergent network morphology. I could have made this whole point the other way around. What kind of network morphology I have shapes what a motor can do. So as a simple example, think of this guy here that wants to walk towards the two plus ends. If it sees two anti-parallel microtubules like here, it will try to walk towards the plus end and make the two microtubules slide in. So these arrows are the direction of motion of the filament, and the filaments will be slid in the opposite direction. Whereas here, if it sees two parallel guys, we'll still link them, but we'll just walk between them without exerting a net force. There's an interplay between network morphology and what motors we have in there that we need to decipher if you want to understand the physics of these things that we see. Before I get there, let me just tell you a little bit, just so you appreciate that this is not like a microtubule only story. This side of the movie, that's again the C. elegans cell that divides. What is labeled here is now not like in this column here, microtubules, but here's another polymer filament, actin, that is labeled, which sits under the cell surface and forms a thin network just under the cell membrane and reshapes it. And so this is sort of a sketch of how you could think about these actin networks. Actin filaments, again, are structurally polar filaments. They have a much shorter persistence length than microtubules in like the tens of microns. So they are, the persistence length, the thermal persistence length is comparable to the size of most cells. So that's why they are called semi-flexible. In the cell cortex, their lifetimes tend to be on the order of minutes. All the relevant processes that we think about are like 30 minutes. So they're again like turning over relatively rapidly compared to everything that we do. Their polymerization, just as the one of the microtubules, consumes energy. So polymerization is an active process. And there's motors associated to them. Well, this movie looks much flimsier. This is actual experimental data. This is a motor that attaches to actin, showed in high-speed AFM as it walks along the filament. So these motors are called myosins. And now that you're primed by the kinesia movie, you can again, once this loops, sort of recognize the structure that you've already seen in the animation from before. Movie ever loops. Of the little guy just taking one step after the other. Back. And so just to give you a sense of this network, so this is now, again, a view of the C. elegans cell that I showed you before. What you see here is the mid-plane view that you already saw before, where you can appreciate the shape deformation that happens during cell division. In the middle, you see the molecular motors where they are on the surface. So this is a surface view. And here you see the filament network of actin filaments. And the point that I'm trying to illustrate here is that this filament network is really essential for deforming the whole cell. So again, this is a physics that is very important to understand if you want to understand life. And I just put this here to sort of emphasize that what I'm showing you here is a very, as I've showed you before, like the examples are really shaped by the flavors of cell biology that I like, but there is, not that I don't like this, I just didn't work on them. But there is like a whole zoo of motile cells. So these are fish keratocytes that are important in fish wound healing. This here is a white blood cell that chases a little bug that it tries to eat. So they're important for the immune system. And I think the number of examples where these cytoskeletal networks really play crucial roles for our life is relatively large, and I don't think we appreciate all of them. Okay, but at the end of the day, like if you just remember one sentence, the cytoskeleton is a set of biopolymer filaments that are cross-linked by motor molecules. Those constituents are highly dynamic, and we want to understand how they work together. I think that would be a good point if you want to have questions about sort of the biological setup to ask them. Otherwise, just keep interrupting me along the way. That is also fine. Okay. So now yeah?
A similar form.
So there's also myosin motors exert forces on actin filaments. It's also a low Reynolds number system. So the total force from the sum of all the forces on each of the filaments has to instantaneously balance. There's no inertia in this world that is appreciable. There's something that I put under the rug. There's, of course, other cytoskeletal filaments, so-called intermediate filaments, that are much more long-lived, can provide like an elastic mesh in which all of this is embedded. So that's how you get something that has a time scale, but inertia will not be it at these lengths. Yes? So in eukaryotic, so in animal cells, you need definitely both. You need actin to sort of divide the cell, and microtubules to tell it where to divide and to move the DNA to both parts. Plants do it different. There, basically, microtubule does everything. In the other realms of life, so this is somewhat specific to where exactly you ask the questions. I think the thing that makes animals special is that we use actin a lot. So for us, we need both. Plants like microtubules better for some reason, but I don't think that there's a generic answer to this question. Okay, so just to put my head as a physicist on now, so the question or the type of system that we want to describe is like a polymer network. The polymers are linked at point of close proximity, and what makes this a new problem is that these cross-linking molecules consume energy and do work inside the network. That is really the setup. At this stage, I would at the screen. Can I? No, I would like to go to the board at this stage. I can do this. Or just use the board on the side. That's also fine. Ah, there's some arrows. Let's try these. Ah, that was easy. Oh, that's fine. Ah, that seems to work. Okay. So basically, what we would like to be able to do is to sort of bridge the scale from, on one hand, so these will be my polymer filaments. They will look much simpler than on the slide. On one hand, filaments that interact via molecular motors to one of these whole networks. Filaments can sort of derive or be able to link motor properties that are defined at this scale to material properties that we could measure in a rheology experiment or something that we could have in the lab. That is where we want to go. And the first thing that we will have to sort of decide so basically, if we look at one of these networks, what we would want to be able to say is if I have a distribution function psi that tracks the position x and orientation of each of these filaments, I would want to know how this evolves in time. So psi would be defined as the sum of some delta functions where each of my filaments here has associated to it a center of mass position xi and a unit vector that tells me the direction in which it points pi. Is that okay so far? And so really what I'm after is dt psi, which you will easily be able to convince yourself this. Will obey an equation like this, where there's really two unknown quantities that are now introduced. This is the flux in position of the filaments. This here is the flux in orientation. At the end of the day, just naively, if I want to know the time evolution of such a network, we'll have to be able to solve for two vectorial quantities, the flux in position and the flux in orientation. So I will have, I will need, oh, there's no, sorry, I. So with the little index I always mean a particle index. If I leave the index away, I mean a coarse-grained quantity that sort of is on the network scale. So I have two of these quantities that I will.
Again, none of this has to do with the thermal. This is all active motion.
Well, in this language, I'm not committing to it, but I will go to a system where basically everything comes from the active motion. The same kind of language can be used for passive systems, and I could refer you to the books by Doy Edwards, where I think this is spelled out in much detail. Okay, so I have two unknowns, I need two equations, and I basically already told you one of those. It's that the force on each filament is always 0. The second equation is that the torque on each filament is always 0. It turns out these two statements are sufficient to derive expressions for these fluxes. Everything that follows now will be sort of rewriting torque and force balance. Okay. Before I do that, let me introduce a little bit of nomenclature, which I will put over there, I guess, so it can stay on. For the coarse-grained variables, I will define a couple of fields in terms of which I will want to describe the system. The first of which is the density, which is just the center of mass density of this guy. So this will just be the sum of all my center of masses, which is also the integral over all orientations of psi. So that is clear. The second one in these networks that can be important is a vector that I will call the polarity. And this vector here goes from value 0 to 1. It will be 0 in a system where everyone points in a different way. So if I have a network that looks like this, P equals 0. If I have a network that looks like this, P equals, in this case, the unit vector in the x direction. Just so we're clear what we're meaning. The next quantity that will turn out to be important is the nematic tensor, which I define just as the second moment. One caveat, in the literature, you will very often find a version of this where the trace has been removed. Here, for simplicity, I'm not doing that. But just be aware that, like, if you read any papers, you will find both versions, and be careful what you're dealing with. This tensor here, in an isotropic system, so again, everything points each way. Q will just be zero. So in an isotropic system, this will just be a trace. In a system that is fully polar, or in a system that is fully antipolar, I guess I should make 4. This guy here in this example would be, if this is my x direction, would be 1, 0, 0, 0, 0, 0, 0, 0, 0. So this nematic tensor tells me something about how ordered, how actually ordered my system is. Just one thing to note about these guys: the polarity and the nematic tensor are in some sense independent quantities. So you can construct a nematic system that is not polar. So this here would have polarity 1, this here would have polarity 0. You can also construct a polar system that is not nematic. So now I am not nematic. And let's put one more so that it's clear. There's no special axis here. But all my arrowheads point towards the right and not the left. So now it would be polar, but it would not be nematic. So these are not quantities that are interchangeable. They have different information. And while very often it is a good approximation to say that Q is just P times P, it is not something that you're guaranteed to have. In general, you can continue the game and now define a third order and a fourth order tensor like that. The one that will come up in my notation is this one, which is the fourth order one. I'll just write it here without giving any further explanation, except telling you that you can play this game for as long as you want. And what you will notice in the cosine theory is that at some point you will need to do what is called a closure approximation to truncate this hierarchy of order parameter fields. Okay, so these will be the quantities that we worry about. And so the next thing that we worry about is the flavor of equations that we get out of there. So even without now putting anything into these equations here, we know some basic physics of just any system that has a conservation law. And the first one that we know is that the density needs to obey a continuity equation. So this can change by a flux of density. And if I don't have any creation or destruction of new filaments, that is it. But in general, I can have a source term here, which would tell me about new filaments being created or old filaments being destroyed. What I did in doing this here is that I introduced one new field, which is the velocity. That is in the way I defined it here, implicit to the thing, the velocity of the center of masses of microtubules. So maybe I should put this in my definitions of microscopic quantities somewhere on this side. Basically, B is defined as 1 over rho x minus x. That is the vectors. So that is the velocity field that came in here. So that is a conserved quantity. The second thing that I know is that this thing here is a conserved quantity itself because it has something to do with the momentum density of the system. So that will only change by a flux. And first term, this flux here is what is called the stress tensor of the system. The flux being the source, the stress being the flux of momentum in the system is introduced in that way. And so what will be the rule of the game is to ask how the stress here depends on these large-scale quantities that tell me something about the local state of my network. That is where we want to go. Any questions about this setup so far? Yes.
Making assumptions about spatial dimension, because obviously these things can be 3D or kind of quasi 2D.
So far I didn't make any assumptions.
Okay.
So this is mainly conservation laws. That is true whenever there's. I think in what I'm doing today, I don't think I will have to do that. Okay.
Another question?
Yeah.
So from the assumption that the force is not equal to 0, it's a bit like putting an acceleration to 0 somehow.
Exactly. So the acceleration.
In the field equation, you get back some kind of acceleration. You have the derivative of the velocity. Velocity.
If I go to 0 Reynolds number, this will become 0 equals. Yes?
In principle, in the flux, or where you have sigma, you also have the velocity.
Sorry?
In principle, when you write dt rho v alpha,
Yeah.
it's not divergent sigma, it's sigma plus the v alpha plus the...
Yeah, this is the total stress, and what you would then do, that you could say that this is some kind of deviatoric stress plus some hydrostatic plus some flux, that we define it with, plus some advection of stress, right? This. Yeah. And again, this guy here that you're reclaiming at low Reynolds number, I will be forgotten. That is the. Anything else at this stage? Okay. Okay.
E alpha, sigma, alpha, beta. The one row up.
Yeah, this is a derivative. Did I do anything wrong yet?
Beta, right?
Yeah, this is the physicist convention, as opposed to the engineer's convention. Yes, you're correct. Turns out this thing will be symmetric, so there's some leeway. Okay, so what we really want to do is, out of the condition that the total force on each filament is 0, obtain an expression for the stress. So in order to do that, I will now have to write down for you what the total force on a filament looks like. So imagine one of the filaments that I care about immersed in a sea of other guys. I want to write down the total force on filament I. Total force on filament I comes from two sources. On one hand, this whole network of filaments is immersed in fluid. So there will be something that looks this. And I give this friction coefficient here an index I to hide all the P dependence on that would make all of this more complicated without making it more insightful. And there's something that comes from the crosslinks. And the way I want to describe what comes from the crosslinks is that I can, in principle, interact with every other filament from any position along the filament that I'm on. So I'm introducing, so this here was the center of mass position. We'll introduce an arc length coordinate, SI, where this position here is xi plus si pi. And that is where my motor attacks. Then it picks filament J. Here I have some xj plus sj pj, the position of the second filament. So what that means is I can have crosslinks that come out and go out from anywhere along this ice filament here. They can grab onto anywhere onto the Gates filament. But there's an extra condition, which is that, of course, while I can reach the guys that are close enough to me, I cannot reach the guys that are infinitely far away. So what I now introduce is a third integral over a ball that is centered around the position on the I filament, that is centered around this filament, and has a radius that is given by the maximal extension of these crosslinks. And I only count my connections if sort of this is geometrically possible given the extension of the crosslinks. And formally I can write this as J bit I Sj minus Sj Ej F I J. Okay. And so that is what I claim is the force of my crosslinks. And this Fij here, which has now the dimension of a force density, will be where all my motor forces, where all my force rules of the motor forces will go in. Is this structure clear?
Is there comment saying what is S means?
Okay, so each of my filaments is a segment, is a line segment. Xi is its center. Si is the distance from its center along the line segment direction at which the crosslink anchors. That makes sense? Si goes from, if this thing is L long, it goes from minus L half to L half to go over the whole filament.
For balance, or is it like Fi is equal to?
The force balance is equal to 0. Now it's the force balance.
So both sides are equal to 0.
So at the end of the day, this whole expression here has to be 0. That is the relevant force balance here. Yeah, this is a plus. I'm very sorry. That is important. So this is what I claim is the total force on each of these filaments. And even before sort of worrying about how Fij now is supposed to look like, you can already see that there's two contributions here. Yes?
Yeah. Here you're assuming you have a... So Fij depends on the distance between the walls, right? Or
so for now I will only, so Fij is minus Fji is all we used so far, but
it will be a function of Si and Sj.
It will be, so it will end up being a function of Si, Sj, and most importantly of the relative velocity between the two attachments.
Velocity, okay.
Yeah.
And then you assume here that you have a uniform density of these forces. So you're discounting the fluctuations related to the fact that you have a number of motors, and so...
Yes. So this is very mean field. The reason why, or the limit in which you get a reasonable result out of that is when you're interested in processes that happen on timescales that are long compared to lifetimes and attachment times of all the filaments. So you have like several generations of motors binding, unbinding, and length scales that are large compared to the filament. So we will not get the fluctuations out of the way that I'm doing that. And I think that's a problem that's, at least in my hands, still unsolved to do that correctly. What helps us is that in the systems that we're interested in, typically at each point in time, you have each filament coupled into the network by some 10s to 20s of motors at each time. So really, like in the spindle, like basically every attachment point is almost always occupied. So yes, this neglects fluctuations, but for the processes that I will use this for so far, we hope that this averages out.
Indeed, that these filaments were bent, and you say they bent due to the forces. So this is totally neglected here.
Yes, my filaments are stiff line segments.
And a related question that I'm thinking now is this bending. So if it bends, it means that there is at the molecular scale something that allows this bending. So is there? Do you know of the barrier energy or limit stress, limit torque or flexural moment that's?
So for microtubules, it is measured. We should discuss it after. I should point you towards the right literature, because I don't want to say anything wrong now, especially since it is recorded. But for microtubules, this is definitely known. For actin filaments, they are much more finicky to get these measurements out. There's persistence lengths, but how much they really answer your questions here is unclear to me. Okay, but next thing what I wanted to try to convey to you is that here in this structure, you see there's already two flavors of forces. One comes from the interaction with the fluid, and one that just comes from the crosslinks, that act on the system. And it's obviously natural to now ask which of these two flavors of forces will dominate the large-scale behavior of the system. Because you could think of a system where basically you rarely encounter another filament, and you do a little slide, and over large scales, all I see is the fluid, the motion that is induced in the fluid. Or you could think of a system where basically everyone is always connected to everyone else, and then I can more or less forget about the fluid. One can make this argument quantitative, but I'm just looking at the clock and seeing that I take way more time than I anticipated. So I will just give you the buzzword to look up. There's something in the fluid, something that's called a permeation length, which is the ratio of the viscosity of the fluid and the effective friction with the microtubule or with the filament network. And the effective friction with the filament network goes up linearly in the density of how many filaments are around. So this permeation length goes down as I get denser. On the other hand, there's also a permeation length that will come out of what I will show you about how far momentum is transported in the gel, which depends again on the viscosity of the gel, which goes up with the number of crosslinks, and quadratically in the density of microtubules, because I have quadratically more possible interaction sites as I have a denser system, and the same friction. So that length scale goes up. So in the highly cross-linked limit, this term here completely dominates the physics of this one. This is the limit that I will deal with now. In the sparsely cross-linked limit, as I want to call it, this term here dominates the long-range dynamics. And I suspect that Andreas Zörtl will tell you a lot about that next week. So for our purposes, this term here is gone. Yes? So repeat the question a couple minutes later, because I will now tell you about Fij. I will try to mention your point, but if I forget it, hold me accountable. Okay, so basically what we end up with is that this sum here, the total forces from all the crosslinks, has to instantaneously balance on each filament. That is where we progress to. So now it will be crucial how we think about Fij. And the reality of the matter is that in cases where many crosslinks connect the filaments, we don't have a very good quantitative answer yet. But what we do know that experimentalists can measure is curves like this. What they do is they take two filaments, they pull on them with a controlled force, and they measure the velocity at which they move against each other. And so this is a very simple experiment, right? If I did that just with my hand relative to the blackboard, you would of course expect that there's a force that is proportional to the relative velocity of my hand with respect to the blackboard opposing it. Just simple friction force. You would expect a curve like this. It would be the force velocity curve, just passive friction. On the other hand, here my crosslinkers move along filaments. So what experimentalists often measure, something that, maybe this is the point in time when a second color, or active red, a curve that then looks like this. The crucial point is that the zero velocity point, this one here, is displaced from the zero force point by a quantity, and this here is what I will call V parallel. That's the preferred speed of the motor molecule that I'm thinking about. To cast this into an equation, I can write this like this. This here would be a force. So this here is the speed in the lab frame of the attachment point on microtubule I. This is the speed of the attachment point of microtubule J. So if I just had these two terms, this here would be like passive friction with a friction coefficient gamma. What is different now in my active system is that the attachment points move by themselves at a preferred velocity V parallel along filaments. So I got these two, now I have red. This is what will make this active. That is the flavor of force rules that we will use in this equation to describe molecular scale motors. So unlike what was suggested earlier, we are not thinking about them as little elastic springs, where the force would be proportional to the distance between the two springs, but as little motor dashpots. The force is proportional to the relative velocity between the two attachment points. We're placing ourselves solidly in the viscous limit of this, again in the long timescale dynamics, and you can actually get at a rule like that if you start with little springs that bind, unbind, and you ask for the forces on timescales that are long compared to the attachment, detachment time. You will have like a viscosity, a viscous force rule that comes out of that on long timescales. One thing that I need to still make this rule work, to project it onto a vector that is parallel to the connection between the two filaments. That is so that there's no net torque on the crosslinker itself. Otherwise, there will be a net torque generated there, and you will end up breaking torque balance. Okay, and with this, the setup is complete. I think the last thing that I owe you is to just at least give you a flavor how you get from such a rule to something that gives you a stress and ultimately the whole dynamics of the system. Basically how I get back to my stresses here. So basically, a nasty calculation that I will not make you suffer through is, and of course, now that I know that Fi, that this here is the force on each microtubule, define a force density, which is this thing here. That's just a definition. And since this comes from something which has the property that Fij is minus Fji, I will be able, after a Taylor expansion, to show that I can write this as the divergence of a stress tensor. And since each of these Fi's is 0, this whole thing here is 0, I will end up with an equation of motion, which is just divergence of the stress is 0, and the stress is defined by plugging the force rule that is here in here, Taylor expanding and keeping lowest order terms in an asymptotically consistent way. But that is basically all that goes into this type of theory. And that is sort of the key assumptions here. And the reason why this is important for us, to be able to do this here, is we can go from a force rule on filaments to a stress. Is there questions? I'll give you the executive summary with slides now again, but I feel this is quite technical, so doing it twice might not be the worst idea. I did not answer your question, that is true. So you asked me, how do I think exactly. How do I think in terms of this force rule here about concentrations of molecules and something like this? What Fij is the expected force density between 2 points of filament that I link. And what I can do is scale this whole thing with the concentrations of molecules that I expect at each point. So I can, on one hand, make these coefficients here themselves dependent on the position along filament. If, for instance, I have a molecule that likes to bind plus ends but doesn't like to bind minus ends, I can make this and this function of SISJ, and this here, SISJ. So that allows me to incorporate like different concentrations along filaments. But I can also globally scale this if I'm interested in concentration gradients on length scales that are large compared to the filament. Does that help you? I would not if I believed that, if I believed that the force rule for 2 crosslinkers is the same as for 1, like if they don't interact, this would be the right thing to do. Now, whether in biology they don't interact, that is what I'm trying to say. In our hands so far, we just add the actions of crosslinkers and everything stays nice and linear, and our coefficients don't change in a complex way as a function of concentration. Biology is more complicated than that. You can have crosslinkers that bind other crosslinkers and prevent them from doing their job and things like that. And then obviously you would have to, if that was important, you would have to engineer this complexity in here. But we haven't played these games yet. Okay, so yeah, this is back. That was over there, right? Okay, so this is what we just did. We used the fact that the total force and the total torque on each of these filaments adds up to 0 to, in some abstract way, or at least I showed you how you would use that to get towards x dot and p dot. What we used for that is that the total force on each of these filaments has 2 contributions, one coming from crosslinks, one coming from fluids. And we argued that there's sort of two regimes of the system. One where I have like filament pairs, where filaments encounter each other rarely, and I see what's further away through the fluid, just a sparsely cross-linked regime, where like it's really a fluid mechanics problem. And one where it's determined by the crosslinks, because basically crosslink dynamics completely dominates the momentum transport. I argued that I would think about these systems, remove the fluid forces, in which case the only thing you had to worry about is something like a force velocity curve between your filaments, which can be generalized by making the coefficients in here position and concentration dependent, as the question pointed out correctly. And just to belabor this point of concentration dependence, we actually worked out, or of position dependence, we worked out sort of the different stereotypes of behaviors that you could have. You could sort of think of each of these crosslinks as something having two heads. They might either walk at uniform speed anywhere, or sort of accelerate near one end, or slow down, or whatnot. Or you might have something that likes to bind the plus end, not the minus end, and walk at the same speed everywhere, or combinations thereof. In this way, you can sort of define 5 flavors of motor heads, connect them in all possible fashions, sort of generate all the force rules for all types of crosslinks that you might think of. And you can work through the whole mathematics, and we have done it for you, and Jakob, who sits there, is the one that you should bother about it. Because he has done the latest version and latest and greatest version of it. And in this way, you can describe all kinds of crosslinks in the viscous regime. What I didn't show you is what you actually obtain after you go through all of this. This is the flavor of the stress tensor that you get out of this. So in our flavor, this whole stress tensor will be proportional to the density squared of filaments, because it will be proportional to the number of crosslinks. And this is in the regime where you're limited by binding sites, not limited by concentrations. There's a viscosity tensor, which is symmetric. It depends on this S that I showed you later, but it is symmetric. And depends on parameters like the length of the filaments and the size of the motors. What is the new physics that you get out of there is that you don't only have a viscous stress, not only something that is proportional to gradients in velocity, but this is now offset by what I want to call an active sliding stress. So V parallel was the preferred velocity of my motors. The effective viscosity is offset by gradients, by V parallel times gradients in polarity. So basically, unlike a passive system, where the force-free situation would be that these filaments don't move with respect to each other, here the force-free situation is that all filaments slide past each other at exactly the speed that the motors would want to slide them. That is where this offset comes from. The second thing that happens if you introduce asymmetries of motors between top and front end, that you get active contractile stresses. You get some that go in polarity squared and some that go into the nematic parameter. So this is actually what you get out of that. The beauty of this is that you can link the exact form of these parameters here to the microscopic details that you put into the system. Okay, so does that work? This was the first experiment at which we released that. There we took a molecular scale motor called XCTK2 that had been characterized 2010 in the Surrey lab. And what you need to know about XCTK2, it likes to bind between microtubules. It binds basically anywhere with the same likelihood, so it doesn't really care where it binds. And at high enough concentration, it slides them with respect to each other at 19 nanometers per second. And so what Bez and Peter, Bez was at that point a grad student in Zvonimir Dojic's lab, and Peter was with Daniel Needleman at Harvard and Brandeis, respectively, built a system out of stabilized microtubules, so they don't grow and shrink. Use a drug called Taxol, doesn't matter. XCTK2 and ATP. And then they squirted this into a flow chamber. And they get this gray gel of microtubules. The dimensions here is these are about 300 microns. This direction is infinity, and out of screen is about 20 microns or something like that. The filaments themselves are 3-ish microns long. There's a large separation of scales. And so if my theory is worth anything, I should be able to tell you how any filament in this network moves. So this equation you've seen a couple of times by now. The nice thing is for XCTK2, we know that gamma is a constant. It doesn't care about SI as J, where you bind. V is 19 nanometers per second. That's all the things that I need to know. The stress then simplifies to just this expression here. And we can basically just see by eye is that this is solved by V is minus V parallel P in stress-free boundary conditions. What this means, we can also put this back into the original force balance, that in this system we expect every filament to move in the direction in which it points at 19 nanometers per second. That is, and that was actually a surprising result for us, because when we thought about filaments originally, would have thought that anti-parallel guys can slide with respect to each other, while parallel guys would just, the motor would just walk between them without inducing any motion. The reason why this result can be rationalized is that we are here in the highly cross-linked limit. So the length scale over which local perturbations are seen in the fluid or in the gel are large compared to the system size. So that means that anti-parallel regions somewhere in the gel can move parallel regions far away, which are still cross-linked, so they still have to move at the same speed. But basically, you have now two anti-parallel gels that just permeate through each other. So far, the theory. So that's the experimental system. We wanted to figure out whether this was true. So what Baz and Peter came up with is that they took a laser and just bleached stripes into here. And you see that these stripes split up as you follow the movie. And if you look closer, you will find regions where these stripes split parallelly, so you have as much stuff going left or right, or regions where basically everything goes, in this case, to the left. What we did is how much stuff goes to the left or right. We took this as a signature of the local network polarity, and we just measured the speed of the stripes. If our theory was right, we should be able to repeat this experiment at different motor concentrations, and regardless of the local polarity, always find 19 nanometers per second. This is the experimental result, 19 nanometers per second. Different motor concentrations, we were very happy. Just so you know that my assertion that this dynamic polarity that we observed had something to do with network structure, we also used a technique called second harmonics generation, which allows you to directly infer the polarity of the system. And you can see that sort of the polarity from the bleach mark and the polarity established from the second harmonics generation nicely fall on a line. So I'm not cheating here. But that means, at least for this simple in vitro system, we're now confident that this theory works. Sort of the last thing that I want to show you is there's also good reasons to think that it works for these guys. These are frog spindles that you saw before. Just to remind you, these speckles here were marks on individual microtubules. You saw all of them move. Their motion depends on a motor called EC5 kinesin. And EC5 kinesin has been reported to move at about 2.5 microns per minute. What we also know is that these spindles are highly polar near the poles, so they're basically all the minus ends stick out. They have parallel regions in the middle. So if this were a highly cross-linked network, then all of these dots should basically move at 2.5 microns per minute. It took a few years, but in 22, the group of John Bruges found exactly that. What you see here, so this goes from the middle of the spindle to the pole, so that's what this axis goes, here from one side to the other. What is reported here is the velocity, which is a straight line. They normalize it by the average velocity, but it's reported in the paper. It's 2.5 microns per minute. I'm not cheating, I swear. And the second thing that they measure, independently by two methods, basically, is the polarity of the spindle, which goes from highly polar to non-polar to highly polar in the other direction from end to end. So that makes us now believe that indeed we have something that is, at least has some key properties of actual living materials in our hands. And that's almost on time. And with this, I think I want to stop. Like, if you remember 3 things, cytoskeletal networks are networks of polymer filaments cross-linked by molecular scale motors that are permanently kept out of equilibrium by local energy consumption. That's very, very different material from like a rubber material. By now, we believe that they are highly cross-linked, meaning that you have so many cross-linking molecules that basically all the long-range momentum transfer occurs via cross-links, not via the fluid in which they are immersed. And in that way, they're different from other active matter systems that you will discuss, I think, next week, like bacterial suspensions or things like that, where it is actually the opposite limit, where like each of these units moves some fluid dipoles around it and talks through the surrounding fluid to each other. So they are their own class of material that is subtly but different from other active matter systems. And the physics of the spindle at least seems to be consistent with these theoretical descriptions. And if you want to ask me about things later on, you can also ask me about actin cortices in starfish oocytes and rings in C. elegans, where we are now also relatively optimistic that this is the right flavor of active matter description for the system. More questions?